All right. Hi, everybody. This is a micro seminar for June 2nd. We are welcoming today Jessie Shapiro, who is doing a lot of awesome things. So Jessie started his PhD with Eric Alm at MIT. Then did a very short postdoc with Pardis Sabeti. I don't know how to say your last name, but Pardis. At Harvard, and then started his lab in June of 2013 at the University of Montreal. I'm not going to try to say anything in French, so apologies for that. In a second, after I will throw it to Jessie, but I just want to remind everybody, if you're on the Google Hangout, please mute your microphones and turn off your cameras. And then if anybody has questions, feel free to tweet at microseminar. Or if you're in the Hangout, just we'll wait till the end and take questions, or even stick them up on the chat, and I'll try to keep a handle on it. So with that, I give it away to Jessie Shapiro.
All right, thanks, Dave. This is really fun. I'm pleased to be here, in my office talking to you all. Hopefully, lots of you. This could be the biggest or the smallest audience I've ever talked to, but I just don't know. Welcome, and I'm about to share my screen with you. Which hopefully just worked. And here we go. So as promised, today I'm going to talk to you about trade-offs in the evolution of pathogens within and between patients. So what do I mean by this? Evolution within patients. Basically, if we have a pathogen, so either a virus or a bacteria, infecting an individual, we'll have a diverse population, or potentially diverse. So this is shown by these colors of viral particles here, due to mutation, or combination, or even infection with different strains and the onset of infection. There can be a certain amount of diversity in an infecting population. Then natural selection can either act within a patient to shape that diversity. So selection from the host immune system, drugs that might be taken by that patient, etc. And then on longer evolutionary timescales, in order for a pathogen to be viable, there has to be transmission from one patient to another. And selection can operate on these timescales as well. And in this example, the blue variant is the only one that is viable to be transmitted from one patient to the next. So this is what I'm talking about when I talk about selection and evolution within and between patients. Which is all well and good, but why would we even care if there's any diversity of a pathogen population within a host? First thing is, if you're an epidemiologist trying to reconstruct transmission chains, it's basically a nuisance. And this was shown in a cartoon by Nick Croucher and Xavier Didelot, where we have phylogeny of a pathogen that's evolving first within person 1 and then is transmitted to person 2. And you can see that it's this lineage here with the red arrow that gets transmitted. However, if we were to sample this genome shown by the dot that has a mutation on this branch from person 1, that might mess up our transmission chain because this mutation is not present in person 2. So only sampling one clone or one isolate of a pathogen per person might mess up the reconstruction of transmission chains. As a side note here, in the spirit of the live tweeting experience, I'm going to try to put out Twitter handles for people that I cite throughout the talk. So this is the first one. All right, thing number 2 about why we might care. Well, if you're an evolutionary biologist like me, we can ask questions about how evolution and natural selection operate on short versus long timescales, and we can ask on what timescales are pathogens measurably evolving. What do I mean by measurably evolving? Well, basically evolutionary distance is a function of mutation rate and time. So if we can measure evolutionary distance, then a pathogen is measurably evolving. And evolutionary distance is a function of mutation rate and the time available for those mutations to happen. So we can break this down. So we've got mutation rate and we've got time that will determine if a pathogen is measurably evolving, if there's a measurable evolutionary distance. So first of all, really simplifying the world, viruses tend to have high mutation rates, so they evolve quickly, and bacteria have much slower mutation rates. So starting with some pathogens that have been pretty well studied that are chronic infections. So these are infections that last quite a lot of time within an individual person. So HIV can last a lifetime. And on the bacterial side, I'll take the example of Burkholderia, but there's a few other examples that I could equally use, Staph aureus, Pseudomonas, tuberculosis, etc. That will be chronic infections over years. So lessons from HIV. In this nice review paper, Pybus and Rambo showed that there are more mutations detectable within patients than between patients for HIV. So here the y-axis is the evolutionary rate in terms of substitutions per site per year. And you can see that it's very low when you compare samples in an epidemic, so that is to say between patients. But within an individual patient, the mutation rates are quite high. And the interpretation here is that within a patient, there's just not enough time for natural selection to purge out deleterious mutations. So most of these mutations that happen within patients are thought to be nearly neutral or deleterious, and that over longer epidemic timescales, they will be purged out and we won't see them anymore. So that's a lesson from HIV. More recently, people started to look at bacteria in chronic infections. And I'm going to take the example of Burkholderia that can persist in cystic fibrosis patients over decades. And Tammy Lieberman and colleagues in a 2013 paper considered two different models of how Burkholderia might evolve within patients. So the first they called the dominant lineage model. And here, essentially, there's strong selection such that all this diversity, so these little branches, get quickly outcompeted, resulting in low diversity within a host because it's basically survival of the fittest. We get recurrent selected sweeps, resulting in a single dominant lineage and very little diversity within a host. It turns out that they actually found support for a diverse community where there are these big purple branches that persist for quite a long time, up to decades, and that in fact each of these lineages is under strong selection and has adaptive mutations that are kind of duking it out in a clonal interference kind of scenario, where each of them do have adaptive mutations, but they persist because they're competing with each other and no single one is able to outcompete the rest. So that was the model that was supported in that 2013 paper. I should say that Von Cooper and colleagues have found similar kind of dynamics also in Burkholderia, that in fact there is strong selection and there can be some adaptive mutations that contribute to within patient diversity. So conclusion of all that is there are some good examples of viruses and bacteria that are measurably evolving over the timescales of chronic infections within a patient. So the question that I'm going to address here is, well, what about more acute infections last much less time. So for example, Lhasa and Ebola virus and Vibrio cholera, where infections aren't on the timescales of months or years, but really on the timescales of days or maybe a week. So, is there any contribution to these infections from diversification within hosts? So I'll start with the viruses, where mutation rates are higher, so it seems more plausible to expect some within host diversity, and then later on I will switch to cholera as an example of a bacteria with a lower mutation rate. All right, Ebola and Lhasa. Normally when I give this talk live, I'll do sort of a poll, how many people have heard of Ebola, how many people have heard of Lhasa, and everyone's heard of Ebola, or I hope, or you should have, and hopefully a lot of you have heard of Lhasa virus as well. But if you haven't, here is a quick primer. So Lhasa virus, like Ebola, is a BL4 biosafety level 4 pathogen, so very, very deadly, not very treatable, no vaccine, with very high mortality rates, so you have to work with it in these spacesuit kind of very, very clean conditions. What makes it quite different than Ebola is that it actually has a well-characterized animal reservoir, so this little rodent, Mastomus natalensis, that tends to live, with humans. It likes to live in people's houses, so it's a little house mouse in rural settings, and it's endemic in large parts of West Africa. So it's easier to get a lot of cases of Lhasa virus than Ebola because it's sort of endemic in a large region, unfortunately. The virus itself, so it's super deadly, just 4 genes, so a 10 kilobase genome. You can see here there's 2 strands. It's a single-stranded RNA virus genome encoding 4 genes. So the nucleoprotein that coats the genome, it encodes its own polymerase, the L gene, a very small zinc binding protein, and the biggest gene, no, sorry, not the biggest gene, another gene, the glycoprotein GPC, that that coats the viral particle. So just 4 genes, and extremely harmful to humans. But I should say not apparently harmful to the rodents that serve as the reservoir. Now, this project was a big one. It was a big collaboration between Harvard, the Broad Institute, as well as collaborators at the Irua Specialist Teaching Hospital in Nigeria, and the Sierra Leone Ministry of Health and Sanitation. So this was work I did with my postdoc, and I'll especially shout out to Pardis, who led the work, and also Christian Anderson and Chris Matranga, 2 other postdocs who I worked closely with on this project. So what did we do? Basically, we sampled clinical cases as well as rodents infected with Lhasa in West Africa. So these are our 2 field sites shown with the yellow stars. So one in Nigeria, one further west in Sierra Leone, and you can also see that this overlaps with the in red circled here the 2014 Ebola outbreak as well. So basically what we did is we got these clinical samples, so either from blood from human patients or from rodents. We sequenced directly from blood, so this is sort of a metagenomics kind of approach after subtracting all the human material, and it was Chris Matranga who really figured out those sequencing methods. We built a phylogeny of all of the reference genomes that we were able to reconstruct for Lhasa virus across all of these samples, about 200 samples in total, and used a molecular clock to date how Lhasa had spread across this region. So this is sort of a big story that is published, so you can read all about it. But I'm going to sort of give an overview and focus on the intra-host evolution aspects. So very quickly, the molecular clock that we used inferred that Lhasa has been circulating for quite a long time in West Africa. So a last common ancestor at least over 1,000 years ago in Nigeria, probably longer, which then spread west across West Africa, getting into Sierra Leone more recently, about 150 years ago. Now, the molecular clock works pretty well on longer timescales, so decades or hundreds of years. It's quite well supported. There's a structure of time in the phylogeny. However, if you look over shorter timescales, it really breaks down. So here, each point is a Lhasa virus sequence sampled over 1 year. So from January to December of, I think, 2011. I don't exactly remember, but I think it was 2011. And then the y-axis is the root to tip distance. So basically, a measure of evolutionary change in a tree. And under a molecular clock model, there should be a positive relationship here where mutations should accumulate over time, and the root to tip distance should be proportional to time. As you can see, it's not. There's really no relationship at all. If we look at Ebola, so again, sampled over a 1 year period, we do see this nice molecular clock where we get a positive relationship of root to tip distance in the tree, so mutations in the tree over time. So Ebola is nice and clock-like, and this is consistent with a simple transmission chain. Also note that the scale here is different, so Lhasa is just a lot more genetically diverse than Ebola. So the thing to remember here, the interpretation of this, is that Lhasa transmission is zoonotic because we've got this rodent reservoir, whereas Ebola is transmitted person to person. So here you can imagine there's a reservoir of Lhasa genetic diversity that's circulating in these rodents, and then each of these human infections, so each orange dot here, is basically sampled from this genetically diverse pool, so it makes sense that there's no transmission chain, there's no relationship between these patients, these orange dots. Whereas Ebola, at least in West Africa in the 2014 outbreak, these were cycles of person-to-person transmission. So each dot here, each gray dot, is a person who transmits to other people, and we get this nice linear relationship. All right, moving into the intrahost diversity section. So essentially, from each patient, we wanted to get a sense of how diverse was the pool of viruses within that person. Very briefly, for each person, we constructed a consensus genome for the virus, and then realigned our sequencing reads. So this is basically metagenomic reads of just sequencing all the virus that's in there. And then we call what I'm going to call ISNVs, so intrahost single nucleotide variants, shown here like this, this site that is a T in the consensus genome, and then you can see that there's some variation where sometimes it's a C and sometimes it's a T. And we imposed all kinds of filters to end up with high-quality ISNVs, which I won't get into, but are described in that paper. And the questions we wanted to ask were basically how much diversity is there within an infected Lhasa patient, and how is it shaped by natural selection, or is it? Is it all just neutral variation? So first of all, just plotting essentially the mutation rate, so the number of ISNVs per site that we were able to confidently call. Comparing Ebola and Lhasa, you can see that Ebola is much less diverse. significantly so compared to Lhasa. So Lhasa is not only more diverse sort of globally in terms of its rodent reservoir, et cetera, but it does seem to be more genetically diverse within human patients. And also, if we break it down into human infections and the few rodents, mastomus, that we are able to sample, mastomus infections are significantly more diverse than humans. So this is consistent with these rodents being chronic carriers where the virus replicates for long periods of time and is able to build up a whole bunch of mutations, much more so than in humans, which are presumably more acute and shorter infections. So the next question we wanted to ask is, so we've got some diversity, we have some isonivs, how are these shaped by natural selection? And we did this in the context of the McDonald-Kreitman test. So this is a fairly classic population genetics test for natural selection at the protein level. And the logic of this test is that non-synonymous mutations will, by definition, have an impact on protein structure. Synonymous mutations will not, so will be sort of less important. That's the role of synonymous mutations is, of course, debatable, and many of them will have effects, but certainly not on the protein structure itself. So in the absence of any selection at the protein level, the ratio of non-synonymous to synonymous mutations should stay the same over time. Therefore, this ratio should be constant if we look at it within or between hosts. So to illustrate this as basically a 2x2 table where we're counting mutations either within or between patients and categorizing them as non-synonymous or synonymous. And then we can define a neutrality index, which is basically a ratio of ratios. So the N to S ratio within divided by between. And just as an example, if we have an excess of non-synonymous mutations within an individual, that will yield a higher neutrality index, so greater than 1, which could suggest diversifying selection within a host. So diversifying selection acting to favor the accumulation of more non-synonymous variants within a host. Or having a high neutrality index could also indicate purifying selection between hosts. So here, this would be a depletion of non-synonymous variants compared to synonymous when we compare sequences between hosts. So using this logic, we annotated non-synonymous and synonymous variants for both Lhasa and Ebola. Here, this is the DN-DS ratio. I'll just go back. So, here I'm talking about N and S, which are just the raw counts of non-synonymous and synonymous mutations. DN-DS are the rates, so normalized per site, but it's the same kind of idea. And you can see that within hosts, Lhasa virus has a much higher DN-DS ratio than Ebola. This suggests that Lhasa virus is under stronger or a longer duration of purifying selection. Purifying selection in this case is basically weeding out deleterious non-synonymous mutations, so pushing down this DN-DS ratio. Again, this is consistent with Lhasa virus maybe being slightly less acute than Ebola, and there being more time for purifying selection to act. If we then look between hosts, DN-DS ratios are much lower, and again, this is consistent with purifying selection being more efficient or having more time to act between hosts, which is basically a longer evolutionary time scale. So next, I wanted to ask, how does this ratio, so the neutrality index, the ratio of DNDS within and between patients, vary across Lhasa virus genes? So you will probably not recall, so I'll remind you, Lhasa virus has 4 genes that code for these 4 proteins that make up the viral particle. So we broke down the DNDS ratio by gene within and between patients, and the Z or Z if you're in Canada or the UK gene is not included here because it's very small. So the ratio is not really informative. The thing to notice here is that the only gene with a significantly different DNDS ratio within versus between hosts is GPC. So GPC has a significantly higher DNDS ratio within hosts than between. And what is GPC? Well, it is this surface glycoprotein, and we hypothesize that the reason for this high DNDS within hosts is that it's on the outside of the viral particle, and maybe it's targeted by the host immune system, and that could select for escape variants, so non-synonymous changes to the structure of that protein that will help the virus evade the immune system. So to get at that, we then asked if these non-synonymous iSNVs in GPC might disrupt B-cell epitopes. So B-cell epitopes are parts of the protein that are targeted by B cells. So to do this, we computationally predicted the regions of GPC that are covered in epitopes. Turns out it's about 40% of the protein is predicted epitope. So that's on the graph on the left here. That's sort of our expectation, right? So 40% of the protein is epitopes. So just by chance, we would expect our iSNVs to land in epitopes 40% of the time. What we actually observe is that non-synonymous iSNVs occur in epitopes over 60% of the time. So this is in red, are all non-synonymous iSNVs that land in epitopes, or in gray that land outside of epitopes. So there's a significant enrichment of non-synonymous iSNVs in epitopes compared to our expectation. As a control, we can also look at the synonymous iSNVs. So these do not affect the protein structure, so we would not expect them to be enriched in epitopes at all. There shouldn't really be any selection for that. And that is indeed what we observe, is that the synonymous mutations behave pretty much as we'd expect. They're just randomly distributed across the gene. In the paper, some collaborators at Tulane actually verified some of these epitopes and verified that the non-synonymous iSNVs do in fact change the epitope structure and help the virus escape the immune system. We also found that these non-synonymous iSNVs, even though they seem to be under positive selection for immune evasion and they can rise to quite high frequency within a patient, fail to become fixed or transmitted from one patient to the next. So there appears to be an important trade-off where selection pushes the viral population in one direction within a host, but that turns out to be a bad direction on the long term, where these variants are not able to be transmitted to new individuals. So to summarize, non-synonymous iSNVs rise to appreciable frequency within a host due to selection, but are unlikely to become fixed or transmitted to other hosts. And I won't get into the details supporting that, but you can read about it in this paper. So back to our big matrix of evolutionary distance here, we can add Lhasa virus and Ebola to our list of measurably evolving pathogens within hosts. So despite being acute infections that don't last very much time, they are measurably evolving, and they are even responding to natural selection within hosts. I'll now move on to the last bit. So this is work going on in my lab currently, so pretty new results that are sort of in progress, looking at Vibrio cholera, which is kind of our least likely candidate for measurable evolution within a patient, because this is also an acute infection that lasts a few days, maybe a week, and it's a bacteria with a relatively low mutation rate. So we're not really sure what to expect in this case. What is cholera? So it's an ancient pathogen. Reports go back at least 5,000 years, probably much older than that. And of course, it's still an ongoing global health problem. So this is a newspaper cartoon from London in the 1860s, where John Snow made the famous discovery that cholera is indeed a waterborne pathogen. So we've got death pumping the water for these people getting infected from the water supply. And on my trip to Bangladesh a couple of years ago, here's a boy in Dhaka, pumping water. And even today, if there is no access to clean water, there is a risk of cholera. So overall, this is a pathogen that infects 5 million people a year, kills over 100,000. These are probably underestimates because these things tend not to be reported that well. In Bangladesh, which is sort of the endemic region for cholera, over 50% of the population is probably affected by the age of 15. So it really affects a lot of people, even if it doesn't kill them. And it's a gut pathogen, so it's usually ingested in water or food. Pretty much takes over, outcompetes most of the rest of the gut microbiota, and causes severe diarrhea, which can lead to dehydration and death. So a mortality of over 50% if untreated. Thankfully, much less than that if treated with simple oral rehydration, and this drops to even 1% or a fraction of a percent supplemented with antibiotics. So cholera is treatable, but is not considered eradicable because it's waterborne. It's basically a coastal marine bacteria that has a lot of other niches in the environment and is probably not going to go away anytime soon. So not eradicable. And there's also no good vaccine. There's a vaccine that's maybe about 50% effective and wears off after a couple of years. And a reason for this could be that Vibrio cholera is so diverse, both in the environment and possibly even within an infected individual, that it's unlikely that there can be one good vaccine. So this might have to be a sort of a vaccine cocktail that has to get developed, or an update, kind of like what happens with the yearly flu vaccine. So it's kind of a moving target. So this is an acute infection. So is there any genetic diversity? As I said, there's infections last a few days or a week, and do we expect to see any diversity? So we set out to ask some basic questions. How many mutations are there within a patient, in what genes? Is this due to co-infection, so two different strains from the environment that infect the same person at the same time? Or is it due to mutations that occur actually within the individual, or recombination events, gene transfers, etc.? And similar to what I was talking about in Lhasa and Ebola, is the intrahost diversity shaped by selection at all, or is it mostly neutral? A first answer to this question came from Kim Seed and Andy Camilli, who published this nice paper called Evolutionary Consequences of Intrapatient Phage Predation on Microbial Populations. And the microbial populations they're talking about in this case are cholera. And they began with the paradoxical observation that most V. cholera isolates from a particular stool sample are resistant to phage isolated from the same sample. So this is sort of puzzling. You know, we isolated a bunch of phage and a bunch of cholera that are resistant to that phage. So why is the phage there if the bacteria are all resistant? What's going on? So to be more precise, from a single stool sample, they isolated 269 cholera, 269 isolates of cholera, and 99% of those are resistant to a phage isolated from the same stool sample. They did whole genome sequencing of 8 resistant and 2 sensitive isolates. And we concluded with quite a lot of certainty that all these genomes are isogenic, so they're really identical except for mutations in either OMP-U or its regulator TOXR. So what is OMP-U? It's a surface protein that happens to be an important receptor for several phages. So this makes sense that mutations in OMP-U could cause resistance. And by doing a bunch of experimental follow-up and characterization of these OMP-U mutants, Kinseed was able to come up with the following model that I won't get into the evidence for, but you can read about in this paper. It's quite cool. So the model that we backed out from this is we started with a patient who gets infected by cholera. So these are the bacteria in gray, and there's a phage that either comes along in the infectious dose or is in the gut. That's not really clear, but that phage will go on to predate the sensitive bacteria, so killing sensitive bacteria and imposing a strong selective pressure on any resistance mutation that might arise. So here, this green bacteria here is a point mutation in OMP-U, the surface protein, that will now be under strong selection because it is immune to phage predation. So it will spread through the population, increase in frequency. In the end, it dominates the population. And then perhaps later in time, once most of the phages go away because they have no one to infect, there could be a reversion mutation. So if a couple of bacteria here that mutate back to the phage-sensitive phenotype. And it turns out that the resistant mutation suffers an important fitness trade-off, and this was shown experimentally, is these resistance mutations are not able to transmit to subsequent hosts. So in order to transmit, there would have to be a reversion mutation, because it turns out that this surface protein, OMP-U, is crucial for host colonization. So again, this is an example of an important fitness trade-off where within a host, in this case phage predation, pushes the bacterial population in one direction that is not viable over the long term because they're not able to transmit to subsequent hosts. So there has to be a reversion mutation either in the host or just in the environment, and if not, this bacterial pathogen population is just not going to be viable. So following up on this story in my lab, we wanted to ask more generally, how common is intrapatient diversity in Vibrio cholera? So sampling a little bit more broadly, additional patients to get a landscape of diversity. So so far, what we have is 5 infected patients from Bangladesh and 3 from Haiti, where, as many of you know, cholera was more recently introduced in 2010. From each of those, we isolate 10 colonies, so that's sort of our our sample of diversity within an individual, and we sequenced those 10 genomes per patient. I should also say that all of these 5 patients in Bangladesh were sampled during the same week in 2013. So it's basically the same place at the same time, and ditto for Haiti, same place, same time. Now, I'm not going to get into too much detail on the SNP calling and such, but I would like to highlight one important control that we did, because we're sort of looking for a needle in a haystack. We don't expect a lot of mutations. So what we did is we picked one colony from one of the patients, subcultured it, got 12 additional colonies, and sequenced those. And we don't expect any real biological variation among these colonies, or no real intra-host variation. All we expect is mutations that might have happened in culture or sequencing errors. So this is a control that is not too often done in sequencing studies, but it's particularly important when not a lot of variation is expected. So we get our sequence reads, we do a de novo assembly, so 1 per patient. We map our reads back to that assembly and compare everything in order to get SNP calls, so between patients, and iSNP calls within patients. From the de novo assembly, we can quantify any flexible genome variation, gene gain and loss. And all of this we're going to filter according to variation in the control. So basically, if a gene or a SNP shows variation among those control colonies that we expect to be isogenic, we're going to filter that out as a likely sequence error or culture error. So here is a phylogeny based on our SNPs. So you can see there's a very big long branch between the Bangladesh strains and the Haitian-like strains, so about 1,400 mutations separating them. We're not so interested in that long-term evolution. So we're going to collapse that branch, which is down here, and focus really on the variation between and within patients. So you can see the solid colors are 5 patients from Bangladesh, and just 1 from Haiti. The other 3 collapse down and are all identical. So this is actually 3 patients from Haiti. Here's the scale of the tree in mutations, and the empty circles are just some other strains that we sequenced from other patients as well. So from this tree, using a simple molecular clock, we infer about 2 SNPs per year. This is a bit low, but in line with previous estimates in Haiti. We think it's low probably because we're being quite strict with our SNP calling. So this is sort of on the conservative side, but we think these are really good SNPs. So another thing to note here is these branches within some of the patients, right? We're just taking the example of patient 4 here. All of the isolates are identical except for this one that has 1 mutation. So there's a few examples like this, and these are our iSNPs that we were able to identify. So we identified a total of 8 mutations between individuals, 5 within individuals, and this is how they break down in terms of non-synonymous, synonymous, and intergenic, and the non-synonymous synonymous ratio and the percent of mutations falling in genes. Now, this is all well and good. It's a whole bunch of counts, but what do we expect? So based on just simple simulations of essentially randomly placing SNPs on the genome, accounting for transition, transversion ratios, and things like that to be realistic, we found that we expect an N to S ratio of about 3. So what we observe overall is a little bit high, but not significantly so. However, there is a significant excess of mutations that occur in intergenic regions, 31%, compared to the 12% that we would expect just by chance. And this is consistent with selection acting against mutations in coding regions. So probably purifying selection that, mutations that happen in genes, whether they're non-synonymous or synonymous, are likely to be somewhat deleterious, and these are actually getting purged out over pretty short timescales. Maybe a little bit within patients, but certainly between patients. And these are patients that are only separated by a few days at the same place at the same time. So pretty short timescales. So here are the 5 genes that have intrahost SNVs. So there's between 0 and 2 per patient. For those of you who like to look at this kind of thing, these are the genes that were hit. I don't want to make too much about the functions. These could have interesting functions, but we don't really know. In fact, I think most of them are probably neutral and can be used to estimate the effective population size. So a few months ago, I tweeted out that I calculated the effective population size of Vibrio cholera within one patient and guessed the order of magnitude, whether it's an effective population size of 10, 100, 1,000, or 10,000. And most people voted for 1,000. But it turns out that the estimates are closer to on an order of, well, 0. That's if there's no diversity, which is true of several patients, or up to an effective population size of about 100. I won't get into the details of how I did these calculations, but they're pretty simple. So these effective population sizes are pretty small, on the order of 10 or 100, and this is consistent with a recent population bottleneck, possibly a transmission, or a recent selective sweep that really purges out most of the diversity. All right, just to wrap up, looking at gene presence absence, so moving from the SNPs to wholesale gain or loss of genes. So this is a heat map where in different colors we have our different patients. C is for the control, and as I said, there's about 10 isolates within each of these patients. And the columns across here are genes. So red is a gene that is present, black is a gene that is absent in our de novo assembly. So first thing that I hope stands out to you is it's quite messy in terms of in control, where we expect no variation among the different control colonies. We do see some variation. So that's a little unsettling in that we're not sure if these are basically assembly errors or actual deletions or transposition events that happen during culture. But basically, what we did as a first pass is all of this stuff we basically filtered out, and we filtered out any gene that showed variation in the control, which gives us this cleaner picture, which is a lot nicer. So here we've got no variation in the control, and the only stuff that's left are things that don't vary in the control, so we think are more likely to be real. So just an example, here is a bunch of genes. So the green kind of shades are all of the Haitian patients, and this is a bunch of genes that are Haiti-specific ICE genes. So these are genes in the integrative conjugative element that are present in Haiti, but not in the Bangladesh strains. And this is pretty consistent with what was found by Lee Katz in their comparison of Haitian and non-Haitian strains. Also interesting, so within Bangladesh, so these are our Bangladeshi patients below the control here, you can see that there is what turns out to be a kappa phage, so about 30 kb worth of genes that varies between patients. So you can see that the patient 1 in yellow has it, this purple patient has it, and it also varies within patients. So this guy, patient 3, there are two isolates that have this phage, and then the rest don't. So this looks like true intrahost variation in terms of an integrated phage being present or not. For the rest of the stuff, so all of this, possible variation intrahost, this is still in progress, and we're looking into it to really make sure this is really real or not before drawing a lot of conclusions. So coming back here, well, we can add cholera to our list of measurably evolving pathogens. And more generally, what we can conclude is that even acute infections that don't last a long time can be at least somewhat genetically diverse within hosts. Maybe just at the level of 1 or 2 mutations, but certainly not to be neglected. More interestingly, intrahost mutations may experience selection, for example, from the host immune system or from phages, and that fitness trade-offs within versus between hosts seem to be a pretty general feature that we see both in bacteria and in viruses. So I think that's why all of this is kind of neat and can be important. With that, I would like to thank everyone for listening. I'd like to thank my lab. I'd like to thank the people who funded this work, collaborators in Bangladesh at the ICDDRB and at Mass General Hospital. Particular shout outs to Inez, Eve, and JB, who were responsible for all the great work on cholera in my lab. So thanks again. Hopefully there's a couple minutes for questions. I will go back to, I will unshare my screen.
Yeah, thanks, man. That was awesome. That's the first, I think that's the first human pathogenic one we've had. So I'm reverberating too. So all right. Anybody have questions? All right. So I want to. Am I still? All right. I think I'm good. So question, Jessie. What do you think? There's small effective population sizes within hosts. How do you think that matters with are there environmental reservoirs of Vibrio or anything, or is it kind of just strictly within hosts once it goes epidemic?
Yeah. So I mean, there certainly are environmental reservoirs, and it's been debated what is the impact of those environmental reservoirs, which are very diverse, on the actual epidemic pathogen. And I think the thinking is that the epidemic pathogen is quite clonal, so it goes through severe bottlenecks, and you get these transmission chains, and it's basically the same thing, which does interact with the environmental pool as well. So there's clear horizontal gene transfers, often located in the ICE, so these conjugated elements and things like that. So there's sort of the classic flexible genome that does interact with the environmental pool, but the clonal background of the epidemic pathogen is kind of the same.
All right. Gonna look through. I don't see any questions on Twitter. Any of you guys on Google have anything to say, or?
Yeah, I actually have a question. Actually, what is a protocol? Do you want us to turn on the video, or how do
you can turn on video. That works.
Yeah. Okay. So it's my first time on here. So I'm Peter. I'm actually in Jessie's lab, but I didn't work on this particular project, so I don't know all the details. I'm curious, Jessie. So when you were looking at the 10 different colonies within each patient, I mean, there was actually quite a few regions of variation. I mean, even within the controls. Could you maybe speak a little bit more about if you think that's to be expected, or if that's kind of within the line, or if it's a little bit more than expected. I guess one of the things I thought about is it possible for a patient to be infected by multiple strains? Would that be seen within the 10 colonies at all?
Right. So, right. So I kind of alluded to co-infection as a possible cause of variation, and I didn't really get back to that. But just looking at the tree, you can see here. I'll reshare my screen for a second. That work? Yeah. So I will. Reshare this. Yeah, so if you look at the tree, all of the different patients form monophyletic groups, right? So, patient 4 is here, 2 and 3 are identical, 1 and 5 are basically identical with a couple of intrahost mutations. But if there was co-infection with totally different strains, we'd see the different patients kind of spread across the tree. So it doesn't seem like there is a lot of co-infection of just totally different strains. So it does seem pretty clonal within a host, and then what happens within a host are mutations. In terms of the flexible genome, yeah, so there can be gene gains and losses, but if they're being gained, where are they being gained from? Clearly, there's phages that might be popping in and out. Yeah, gene losses make sense. Transpositions. What we haven't really been able to look at yet is like actual gene transfers from the resident microbiota. And I don't know. It could be, but we haven't been able to clearly see that yet. I don't know if that was your question.
Yeah, that was essentially my question. I was just I guess I'm always surprised seeing that there's so much variation within those 10 colonies, especially relative to the single nucleotide variants that were found.
Yeah. Well, I mean, as I said, if you just from the control. So basically, you start with a clone, you get 10 new colonies, you sequence those, and you assemble genomes and align them. Whether it's from the assembly or from real gene gain loss events that are happening in culture, yeah, there's a lot of messiness. So I think this is an important message is, if you're looking for fine scale variation over short time scales where you don't expect to see a lot, you really do have to do your controls and make sure you trust your assembly and all this kind of stuff.
And I'll just add too, like at least in the bugs I've worked with and I've sampled the diversity in, like you actually get selection for growing up into a colony too.
Mm-hmm.
So I've always, it's always hard to know, like, when did the mutation truly happen when you're growing it up into a colony, because it might just be that one cell, that's the one that grows. So
yeah, that's it.
Yeah, cool. So I don't see anything question-wise on mine. I think I'm good here. My family's running around like crazy, so we're good. But thank you very much, man. That was awesome, and that's really relevant and topical at the moment. So that's awesome. So thanks again. Unless anybody else has questions, we'll sign off.
All right. Thanks a lot. See you next time.
All right, man. Yeah.
Bye.